Hey everyone! In the last video, you learned about the importance of cell cycle checkpoints and what they check for. In this video, we'll discuss the actual mechanism of these checkpoints, in other words, how they work at the molecular level. In this video, we'll be answering two questions. And our two questions are, how do Cdks, cyclins, and other proteins enforce each checkpoint? And how does p53 protect the genome? Last time we examined the various factors, like cell size and DNA damage, that determine whether a cell passes each checkpoint and eventually divides. Some factors, like nutrients and growth factors, push the cell forward through the cell cycle, while some factors, like DNA damage and lack of spindle fiber attachment, pause it. These factors do this by changing the activity of proteins that enforce these checkpoints, usually by causing these proteins to be modified in some way. Two types of proteins that regulate cell cycle checkpoints are cyclins and cyclin-dependent kinases. Cyclin proteins do what they sound like. Cyclins regulate the cell cycle. There are four types: the G1, G1S, S, and M phase cyclins. Their expression, or how much of each cyclin protein is present, changes greatly throughout the cell cycle. When the expression of each cyclin peaks is when it carries out its function of driving the cell into the next phase of the cell cycle. In fact, each type of cyclin is named according to when this occurs. For example, G1S cyclins accumulate just before S phase and drive the cell into S phase and initiate DNA replication. The exception to this is the G1 cyclins, which function throughout the cell cycle. Cyclins accomplish cell cycle progression by regulating the activities of enzymes called cyclin-dependent kinases, or CDKs. CDKs are kinase enzymes. Kinases take a phosphate group and attach it to some molecule, in this case a protein, yielding a phosphorylated protein or phosphoprotein. Phosphorylation is a common way proteins are activated or deactivated. Some proteins are active when they have a phosphate group, while other proteins must have a phosphate group removed to become active. This is a quick way for the cell to start or stop what a protein is doing. These phosphate switches are so central to a cell's life that the human genome encodes 518 different kinases, each with its own targets. Why are they so important? Think of phosphates as a light switch, and proteins as a light bulb. It's easier to turn the bulb on with the switch rather than replacing the bulb or making new protein. It's also easier to turn off with the switch rather than wait for the bulb to burn out or for the protein to degrade. CDKs, unlike cyclins, have a relatively steady expression throughout the cell cycle. So they're usually around. But as their name suggests, cyclin-dependent kinases require cyclin to bind to them to become active. Each CDK has its own cyclin or two that activates it in this way. For example, a CDK that binds a G1S cyclin only becomes active as the cyclin accumulates. The cyclin also guides its CDK to the correct target proteins, so the CDK can phosphorylate them. These target proteins then facilitate the cellular events required to move the cell to the next phase. When cyclin levels are low, the CDK that it binds to remains inactive, and the cell stays in the phase that it's in. This is why high cyclin concentration is required to move to the next phase. When cyclin concentrations are high, there's enough cyclin around to bind to the CDK, activating it so that it can phosphorylate its target proteins and drive the cell cycle to the next phase. Now let's take a look at what happens at the G1S transition. During early G1, the cyclin-dependent kinase CDK2 is inactive because its cyclin E, is present at low concentrations. One of CDK2's targets, the RB protein, therefore remains unphosphorylated and is able to bind the protein E2F. E2F is a transcriptional activator in that it can turn on the transcription of genes, DNA replication genes in particular. However, when E2F is bound by RB, it can't do this, so these genes remain unexpressed, and the cell stays in G1. But by late G1, cyclin E has accumulated to higher levels and can now bind CDK2. This activates CDK2, which phosphorylates RB, causing it to release E2F, which is now able to induce transcription at DNA replication genes. These DNA replication enzymes are therefore produced, and the cell is able to pass the G1S checkpoint and move into S phase. So we see here that it's the accumulation of cyclin E that allows the cell to pass the G1S checkpoint. The G1S checkpoint is also one place that the cell can pause the cell cycle if DNA damage is detected. As you saw in a previous video, DNA damage induces the accumulation of the p53 protein. p53, like E2F, is a transcription factor and binds to genes to alter their rate of transcription. p53 induces transcription of genes encoding DNA repair enzymes, inducing DNA repair. At the same time, p53 increases expression of a gene encoding a cyclin CDK inhibitor, or CKI protein. This protein binds to CDK cyclin complexes and deactivates the CDKs, preventing RB phosphorylation, so E2F remains bound to RB, and DNA replication genes remain off, so no DNA replication enzymes are made. Recall that the G1S checkpoint checks for these enzymes, and in their absence, the cell is paused at the G1S checkpoint. This allows DNA repairs to take place. So, in response to DNA damage, p53 induces DNA repair and enforces the G1S checkpoint via CKI. It also induces apoptosis if DNA repair fails. Together, these functions of p53 prevent a cell with damaged DNA, which could potentially cause the cell to become cancerous, from reproducing and possibly forming a tumor. Sequencing cell cycle genes from tumors is big business in the genetic testing field, because it allows physicians to tailor their treatment based on what genes have been mutated. Mutations in the p53 gene are the most commonly mutated gene among all cancers. This is because when p53 is mutated, the cell loses DNA repair, part of the G1S checkpoint, and it loses apoptosis in response to failed repair of mutations. So cancer-causing mutations can accumulate more quickly without causing apoptosis. This allows cells with a large number of mutations to reproduce. Another CDK cyclin pair is at work at the G2M transition. The kinase CDK1 is expressed at fairly constant levels. Cyclin B accumulates as the cell cycle progresses, reaching high concentrations by the end of G2. During G2, cyclin B binds to CDK1, forming the maturation promoting factor, or MPF, which, like other CDK cyclin pairs, forms following cyclin accumulation. The active MPF now phosphorylates its target proteins, which in turn initiate the events of early mitosis, including breakdown of the nucleus, chromosome condensation, and synthesis of the spindle apparatus. Because cyclin B must accumulate before this happens, the cell is stopped from initiating mitosis too early before S phase and G2 have been completed. So again, we see progression to the next phase of the cell cycle being controlled by the accumulation of a cyclin, in this case, cyclin B. The G2M checkpoint pauses the cell cycle if there is DNA damage. This is because DNA damage inhibits the activation of MPF, halting progression into M phase. No MPF activity, no target phosphorylation, and no mitosis. Okay, so here I'll show you how we think the spindle assembly checkpoint works, which takes place at the metaphase to anaphase transition. During mitosis, if both sister chromatids are attached to spindle fibers from both opposite sides of the cell proceeds to anaphase. This requires that a protein called separase degrades the cohesin holding the sister chromatids together, allowing their separation. Before anaphase, separase is bound by a protein called securin, which stops it from degrading the cohesin between the chromatids. But how does the cell prevent separase from being released from securin when not all sister chromatids are bound by spindle fibers from opposite sides of the cell? It does this through the action of a large complex of proteins called the anaphase promoting complex / cyclosome, or APCC. If all chromosomes are correctly bound by spindle fibers, APCC attaches a small protein called ubiquitin to securin. This ubiquitin tag marks the securin protein for destruction by the proteasome, an organelle that breaks down proteins into their constituent amino acids. The destruction of securin releases separase, allowing it to break down cohesin so that anaphase can occur. Ubiquitination is often used to target proteins for destruction by the proteasome, and as a way for the cell to quickly destroy a protein, in this case, securin. So, okay, this tells us a bit about how anaphase begins if all is well, but how is the spindle assembly checkpoint enforced if something goes wrong? Most genetics textbooks I've seen don't really cover this, probably because it's not really clear what the primary signal for enforcing this checkpoint is. The research data generated to answer this question seems to point to two different proposed mechanisms or models. But we won't let a little uncertainty stop us, so we'll just examine both of these models. Now, in the attachment model, a kinetochore that fails to attach to any spindle fiber sends out a stop signal, which inhibits the APCC and prevents the destruction of securin, keeping separase inactive and holding the cell in metaphase because the cohesin is keeping the chromatids together. The second of the two models is called the tension model. In the tension model, when spindle fibers from opposing sides pull on the chromatids, the pulling force they exert on the chromatids stretches the centromeres of the chromatids apart, which allows anaphase to progress, because there is no stop signal sent out when this occurs. However, it's possible that only one kinetochore is bound by a spindle fiber, as shown here. It's also possible that only spindle fibers from the same side attach to both kinetochores of a protein, as shown here. Both situations fail to create a pulling force on the centromeres of these chromatids. And this lack of tension is what generates the stop signal and inhibits APCC, in turn holding the cell in metaphase until tension is achieved. Both of these models are supported by quite a lot of data, and both mechanisms probably operate in the cell, but it remains to be seen which signal, the signal generated by unattached kinetochores, or the signal generated by lack of tension, is the primary signal that pauses mitosis at the spindle assembly checkpoint. Perhaps this even varies by organism, or by cell type, or by some other variable. The APCC complex of proteins does much more than what I'm showing here, and also functions in other phases of the cell cycle to degrade other cyclins and prevent premature progression through the cell cycle. Indeed, throughout this video, I've shown only a small part of how the cell cycle is regulated, and I've highlighted just a few of the CDK cyclin pairs. What I have shown you is simplified in terms of how the checkpoints are enforced. Also, much about checkpoint enforcement remains unknown, but given the importance of cell cycle regulation in cancer development, I expect that we will continue to gain knowledge about this subject. I'd like to end with a study tip. These diagrams of molecular processes can get pretty complicated, and they take time to learn. When studying, after looking over this material, put it away and see if you can recreate the entire diagram. Write down one component that you know, for example, the APCC complex, and try to remember how that component affects the others in the pathway, for example, securin, and how that component is affected by other things in the pathway, for example, unbound kinetochores. Then try to draw as much of the diagram as you can without looking back. Research shows that drawing something is better for learning it than just looking over it. I'll leave a link below this video if you're interested in that study. This is one reason I have you fill out the learning guide after you finish each video, and why I draw more than I write words. Okay, so at this point, you should be able to explain how checkpoints are actually enforced and how p53 protects the genome. If you don't feel like you can, as always, you can always watch part or all of the video again. And make sure to complete the learning guide as well. Now, in the next video, we'll take a look at the basics of cancer genetics and how mutations to cell cycle checkpoint genes can lead to cancer. See you all then.